Which one's it?
I'm going to go forward, and this is your laser.
Yeah, go ahead and ask a question.
Okay. Talked about killifish and having a small area that they operated in. Could you speak to all the other organisms that we are concerned about and whether they are more typical of the killifish or more typical of animals that can go far and wide and away from the spills if that was possible?
All right, I'm going to relay your question to our audience here, and then I'll take a stab at answering.
What's your question?
The question was, how does, all right, so they understood that the killifish hangs out at one spot for a long time, so it's kind of representative of that location. But what about other animals in the Gulf? Do they hang out at the same location all the time, and can you kind of relate their health to that location? My understanding is that many of the species that we're interested in range far and wide. You know, the tuna, the whales, a lot of our birds, they travel and visit many places. And so it would be a lot more difficult to detect an effect of one of those animals visiting multiple locations across a year or two. So I think those animals it's a lot harder to figure out, to detect an effect. Chris, you got anything to add to that?
Well, yeah, like, you know, one of the things with the open water, you have a lot of species, you know, a lot of high-profile species are going to move around, things like that. And you're right. What happens is you want to look where the oil goes. It's going to be the sediment, you know, other components.
Chris pointed out, for those of you on the line, that in addition to the animals moving around, the oil is also moving around out in the Gulf. And so it, you know, it gets buried in the sediment for a while at a place, but then it can be mobilized after a storm. And the oil's moving, the animal's moving. So detecting the effects of oil and dispersants on animals in the Gulf of Mexico is a very challenging job, and one that neither Chris or I do. We always work in the greenhouse or in the laboratory where we have these aquarium where we can keep the salinity constant for 2 or 3 months while we let the oil biodegrade. And that way we can infer the effects of salinity on these processes. Your question is very important, but one that we really can't answer with the tuna and those bigger animals. All right, I'm going to go ahead and, did that answer your question?
Yes, I think so.
Thank you. All right, I'm going to go ahead and move into...
Are we talking about like all species? Are we talking about things with economic value, or who's looking
Could we
at?
please hold all questions till the very end?
Yeah, we're going to come back to your question. Hold that till after my presentation, and then we'll revisit that. All right, I'm going to go ahead and Chris gave you an example of one experiment, and I hope you got a sense of the amount of effort that goes into be able to say, like what he said at the end, the dispersed oil was more toxic than the undispersed oil, and that toxicity persisted longer. That's so easy to sum up and say, but I hope you get a sense of all the aquarium that had to be set up with the same amounts of sediment. The water's the same except for the salinity. We've got the oil. You got all those fish you have to raise and expose them at the right, raise them at the right age. It's an awful lot of work to say a sentence like that. What I'm going to do is... I've lost my microphone? Well, all right, I'll keep it right by my mouth. Thank you. What I'm going to do now is I'm going to say a lot based on a lot of research, and I'm not going to give you any details on how that research was done. I'm going to cover a lot of ground. That's my intent here. So the background information, what I want to remind you is how many things are in crude oil. Chris said this, and I'm going to say it again because it's extremely important. There are tens of thousands of different compounds in crude oil. I've heard in presentations up to 750,000. I found citations up to 36,000. I'm not sure why I've heard that difference, but there's a whole lot more compounds in oil than we can chemically identify, and certainly even more and even fewer of them we can identify and measure their concentration. And that turns out to be a very challenging part of the problem we face. We can measure about 150 of those compounds. As time goes on, the chemists are getting better at identifying a compound and determining its concentration. We're up to about 150, and you hear this during the oil spill. Ed Overton was on TV a lot, and you hear about oil fingerprinting. This is the fingerprinting. It's GC, mass spec, type work, and they call, I'll refer to it as fingerprinting. It's kind of it's the state of the art in oil spill response, but it's a big difference from being able to fingerprint and say we got these 150 compounds and recognizing that there's at least 36,000, maybe 750,000 compounds out there in the crude oil. The dispersant is a mixture of chemicals. So again, it's not a single compound. It's a whole mixture of compounds designed to break up oil and dissolve it in water. All right, so I'm going to review first kind of an overview of the effects of oil and dispersed oil on our coastal ecosystems, and then I'm going to start talking, I'll move into alternative dispersants. But first of all, I'm going to talk about marsh grass, and then I'll talk about the microbes that live in the mud, and then I'll come back to the animals where Chris was, and then we'll move into the next generation, or search for a new dispersant. Well, why I care about the marsh grasses? The grasses are important because they're the base of the food web. They make, they do the photosynthesis. They make the litter that the worms eat, that the shrimp eats the worm, the trout eat the shrimp. Those are that's the base of the food web. If you kill the grass, you're going to have less energy going in the food web. The above ground parts are structurally important because this is where, again, some animals eat this directly, but also it's important, particularly during winter, to provide cover from cold weather for a lot of wildlife. The below ground parts are especially important. Emily mentioned up front about the roots. The live roots are what give these soils their strength, and that's what makes the soils resistant to erosion. If you kill the roots, you reduce the strength of soil, and you therefore increase the erosion rate. The below ground part, the roots are also important because it builds peat. You think about trees, they grow, they get bigger and bigger around all the time. They lay down wood, layers of wood. You can cut a tree down and see those rings. A marsh is doing something similar, but you're getting sheets of root building up on the marsh year after year. And this is how a lot of our marshes offset subsidence and sea level rise. So if you stress the plants and just reduce the production of roots, you're potentially increasing the erosion rate, and you're slowing the rate at which they build up. You're slowing their ability to offset subsidence and sea level rise. And then the marshes drown and convert to open water. All right. Here's a picture of some roseau cane down at the mouth of the river just during the spill, and you see a bunch of dead leaves. The oil has come in on the bottom of this plant. It appears the oil has been moved up the plant and killed all the leaves in the plant, and all the leaves are dead, and yet you see these new green leaves coming out. I'd seen this in the greenhouse 10 years before with a different species. It really shocked me that there are a lot of marsh plants, a lot of marsh grass, a lot of marsh plants are very tolerant to crude oil. All the leaves die and it just makes new ones. Now they can't do this over and over again. You're just going to run out of energy reserves after a while. But many marsh grasses are tolerant to crude oil. And there's been a lot of work looking at dozens of research projects looking at the effects of crude oil on marsh grasses. And we can go ahead and summarize that. There's too many. I'm not going to go into any specific studies now. But the pattern's clear. The oil on the plant is a whole lot less of a problem than the oil on the roots. The roots are where the plants are uptaking stuff to bring to the rest of the plant. They're also important. They have a hard job getting oxygen down to them. The upper part of the plant basically is a snorkel for the roots. That's what makes a wetland plant. And the oil getting on the roots is a much worse of a problem for the plant than oil getting on the stems and leaves. The other pattern that we also see from looking at the response of plants to different concentrations of oil and then looking at how it happens out in the field is that I now think of it as 3 zones of concentration in the field. This red zone here where there's enough oil in the soil to kill the vegetation and prevent new vegetation from moving in for months and maybe even years at a time. Beyond that zone, you can often find a larger zone where the concentration of oil is lower. The adult plants can survive, but they can't reproduce. And beyond that's an even larger zone, the light green here. Battery's dying. The light green, where the plants have a little stress, but then after a while they do recover and they come back. And that's the way I think about this now. And this you can, well, and it's really all about the oil in the soil. Well, not all about. It's a lot more important where you get oil in the soil than just on the top of the plant. There's been enough work by enough researchers now that we can actually rank the sensitivity of different plant species to oil. Not all plants have the same tolerance to crude oil. Some are more tolerant than others. All right. The map that I'm showing you here is a map of the different marsh types in coastal Louisiana. Marsh type is a group of plants that we recognize that occur in different salinity zones. Like the purple color up there is the bald cypress swamps. You come further out, the dark green color, that's what we would call a fresh marsh. Come on down, there's a light green color. Most of us refer to that as intermediate marsh. Then you get to the red, which is the brackish, and the yellow one is the salt marsh. As you move from low salinity to high salinity, the number of species in the plant community drops tremendously. And by the time you get to salt marsh, there's really only one species that dominates them. When you get up in the fresh marsh, it's very patchy. You've got many species, and you can have, there are several plants that may dominate. One species might dominate here for a couple hundred acres. You move over a mile or two, it might be dominated by a different species. And there's a lot more biodiversity in the fresh marshes. But if we look at the rankings of these, you see that our most sensitive plant species is Phragmites, the one that I showed you. It also occurs at the very end of the Mississippi River. You might think that you look at the map of Louisiana, and that sticks the farthest out in the Gulf of Mexico. You'd think that'd be the saltiest plant communities we have, but that's not true. Those are dominated by fresher communities because there's not enough salt to stress these plants, and the salt plants can't make it up in there. So the Phragmites, some of the first areas that get hit by oil, are also some of our most sensitive to oil. The Spartina patens, which dominates our brackish and our intermediate marshes. Many of you will know it as wiregrass. I forgot to mention the Phragmites is all rosocane. If you build a duck blind, you're building it out of rosocane. That's our most sensitive. The wiregrass, the Spartina patens, seems to be our next most sensitive. And then you get to kind of a tie between Panicum hematomen, pyphene, it's a fresh marsh plant, maiden cane, and it's kind of tied up with the salt marsh plant, Spartina alterniflora, the oyster grass. Many people call it oyster grass because when you start seeing it, you know you're salty enough to find oysters in the water. And our most tolerant species is the bulltongue, a fresh marsh plant that just seems to tolerate crude oil very well for some reason. Notice the fresh marsh has our most sensitive and our most resistant species in it. There's no relationship between the sensitivity of or the tolerance of these species to flooding stress in oil, or between their tolerance to salinity stress in oil. These effects are unrelated, it turns out. And this is useful information. If you're a spill response person, you can use your ability to identify plants to know where to put boom. If you don't have enough boom to cover everything, what do you want to do? You want to put the boom in front of the most sensitive species you have on your piece of real estate. Also might help explain why you might see more effects farther inland than out on the coast. Notice the salt marsh plant is more tolerant than our Spartina patens, our brackish marsh plant. So it's possible to have less oil do more damage further inland because the plant species differ in their sensitivity to oil. All right, I want to move on and say a few things about marsh mud. Marsh mud's really cool. It really, really is. I never would have thought this about 30 years ago, but marsh mud is some really cool stuff. Why? And everyone should care about marsh mud. Why I care about marsh mud? Well, because it rots. Rotting is really cool stuff, it turns out. It's really important in the environment. If any of you garden and you compost, you're using that rotting of that material to release nutrients, nitrogen and phosphorus primarily, for your tomato plants, whatever it is you're growing. Out in the marsh, that same process is what supplies a lot of nutrients to marsh vegetation. So you can imagine if you were to kill all the bacteria that decomposes peat, you're going to reduce nutrient availability to the plants that make the roots that hold everything together. So we don't want to have an oil spill or a dispersant or anything really come in and reduce the rate at which the peat is decomposed by the bacteria. But also, these bacteria are eating this peat. They're eating the thing that builds the elevation that offsets subsidence and sea level rise. So you don't want to speed this up either. You might say, well, let's speed it up and get more nitrogen and phosphorus available to plants. But if you do that, you can actually eat up the thing that supports the surface elevation of the marsh. So you don't want to slow this down either. We want to keep it the way it is. I wouldn't want to change this because I wouldn't know which way it would go. You know, a lot of these marshes are barely hanging on as it is. They're barely keeping up with subsidence and sea level rise. I certainly don't want to accelerate decomposition on these things. These microorganisms that live in the soil also eat the oil. Now, they don't take bites out of it. I'm going to say eat. I do want to make clear what they're doing is they're secreting enzymes that break down the oil into molecules small enough for them to absorb and then eat and get their energy that way. But this is a community that eats the oil. So you don't want to put something in here that's going to kill the bacteria that are supposed to eat the oil, because you could sterilize the very thing that's going to eat all the oil up for you. So again, we don't want to mess up this soil microbial community. Yeah, and that reduces the toxicity to the plants and the animals. All right, so it turns out that when you add oil to marsh mud, it increases the rate at which that soil decomposes. The microbial community gets faster. It's probably a different community than the one that was there. There are probably definitely a lot of microbes that are, I would assume, are stressed out, toxed out. But new bacteria take over and they start eating the oil, and they also eat the peat faster than they would have eaten it without the oil. Our dispersants or fertilizers. Some people want to fertilize the oil slick so the bacteria can eat the oil faster. In the experiments that I've seen, the ones that I've done, these do not reduce this enhancement of eating the peat. How do these things affect the hydrocarbon disappearance? Well, it depends. In one experiment, it depended on which way you measured the oil. Remember, I mentioned that we've got this fingerprinting thing, and they're making it better all the time. There are other ways to measure the oil, too. If you use one way of measuring the oil, you say, oh, the dispersant helped the oil disappear faster. But if you looked at another way of measuring the oil, you go, no, there's no effect there. In the salt marshes, it was pretty clear both way, I can't remember right now. I want to say maybe it's just one way, maybe two ways. I have to go back and remember this paper again. But the dispersant slowed the rate at which oil disappeared over time from jars of mud we held in the laboratory. And this really, I hope this is making you think about what's the best way to measure the oil. You know, what's the best way to fingerprint this stuff? I've got citations at the bottom of here. If anyone wants to take this this PowerPoint afterwards, you can take it and you can track down the sources of my information on these slides. All right. I think the most important way to measure the oil chemically is the one that most mimics what it does to animals. There's a couple reasons why the animals are important. Well, they're ecologically important. Many of these guys are food. Yes? Many animals are ecologically important. They're food or predators or other animals. Many people make their living catching animals. They're recreationally important. They're emotionally important. You know, looking at the birds and the oil spill definitely could get your emotions going. And probably our physiology responds more like their physiology than the plants or than the microbes. Trying to guess how oil and dispersant is going to affect you, you're probably going to want to pay attention to these fish studies that Chris talked about more so than the plant studies and the mud studies that I've been talking about. All right. It turns out that crude oil is not the worst thing out there. I've worked with diesel oil. I've also worked with fuel oil. And these refined products are more toxic than the crude oil. Crude oil is less toxic than things we make out of crude oil. Toxicity declines over time because you've got you've got bacteria out there who can eat the oil. This one's real important to me, the one I just popped up here on the screen. The toxicity of the soil and the water are unrelated. We've seen this twice now in our experiments. Chris described his aquarium to you where he's got the oil and the mud. I mean, he's got the water and he's got the mud below. You can analyze the toxicity of the water to fish, and you can use the mud to do it to worms. And you would hope that where you had your most toxic water to fish, you'd also have your most toxic mud to worms. It turns out that's not the case because the oil is going to different places. Sometimes there's more of it up in the water, and sometimes there's more of it down in the mud. You cannot predict the toxicity of a site simply from measuring the water or simply from measuring the toxicity of the sediment. You need them both. You can imagine how complicated this gets in a coastal estuary where the tides come in and out every day. The toxicity of a site is probably changing as the waters move in and out and bring in different bodies of water. Citations. All right. When we do see differences in the lab between dispersed oil and undispersed oil in toxicity, the dispersed oil has been more toxic than the undispersed oil. Chris showed you one of these experiments. You know, sometimes there was no difference in toxicity between the dispersed oil and the corexit. But almost always the dispersed oil is more toxic than the undispersed oil. But in the experiment, you don't see a difference sometimes. When there is a difference, though, the dispersed oil is more toxic than the undispersed oil. The toxicity is prolonged by the dispersants in our laboratory studies. Chris showed you one example. I had some more graphs I took out of here to save time, but this is not a one-time observation. Seen this in multiple studies. This effect of dispersed oil being more toxic than undispersed oil is greater at lower salinity. Again, think about the very tip end of the Mississippi River. That's where the stuff first, that's where oil, you can see it first come ashore. And that's probably, if there's oil coming ashore, you might have dispersed oil in the waters underneath, also possibly coming ashore there. And in a low salinity environment, being diluted with the river water. And this one's, now I'm wrapping around. Remember we talked about the chemicals. We cannot yet predict the toxicity of a sample based off the chemical analysis of the sample. I wish we could, because the chemical, as expensive and difficult as the chemical analysis are, the toxicity measurements, as you saw Chris describe in one experiment, are even more complicated. I'll show you. Here's a graph. This is from work what I did here when I was at University. We were at University of Southwestern Louisiana back then, USL, UL Lafayette now, with Paul Clerks here in the biology department. And what I'm showing here are two graphs. These are our best relationships between chemistry and toxicity. The lines represent a statistically significant relationship between a chemical that we measured and the toxicity. What you see on the vertical axis is the survival. So higher survival at lower concentrations, and then as the concentration picks up, you see the survival go down. So the concentration of the compounds are on the horizontal axis. But look at the spread of the real data points around those lines. If we're using the chemistry measurements to decide what is clean and what is not, where to spend our limited resources, you're going to get some of them wrong. You're going to end up spending money to clean up sites that really aren't all that toxic, even though they have a high concentration of these chemicals. And unfortunately, you're going to end up not cleaning up some sites that are actually quite toxic because they don't have a high concentration of these chemicals. I wish I knew the answer. I mean, this is the best chemistry we got, but the chemistry does not yet allow us to predict toxicity accurately. This is not just our work. Chris showed you some of that work. Another repeat study, Adam Kuhl was a senior author. There's these two previous studies, there's another one I've seen recently. This is not, we're not the first people to show this, certainly, and we're not the last people to show this, but this is one message that isn't out there. I was at a meeting a couple of years ago. Some people from Coast Guard and NOAA from Washington, D.C. were talking about how they wanted to end toxicity testing and oil spill response because it was not matching what the chemistry told them. And that is bass backwards. The toxicity is what is important. That's, if it doesn't match the toxicity, then the chemistry is not right. It's not the other way around, in my opinion.
What's PTAH?
This is total target aromatic hydrocarbons. We measured hydrocarbons, I want to say 8 different ways in this study, and tried to see which was the best one, and these are the 2 of the best from that study. All right, so yeah, I believe that the best chemical measurement is the one that most closely matches the toxicity, and I trust, we have to use chemistry data, so I trust the fingerprinting over the other things that are out there, such as total petroleum hydrocarbons. There's some fluorimetry measurements out there. The fingerprinting is the best we have, but don't start thinking it is representative of the toxicity. All right. I'm now going to switch gears and start talking about some of the things I've learned about dispersants and surfactants as people have contacted me since the spill, and Chris and I started working with people trying to develop a new and better dispersant. All right. So a dispersant I mentioned earlier is something that we use to try to break up the oil slick, because you want to prevent it from getting to the marshes, you know, like Emily showed you. We want to prevent that. We want that oil to be dispersed, become tiny drops, and get eaten by the bacteria far away from our coastal areas. The dispersant is made up of many chemicals itself. One of the most important types of chemicals in a dispersant is called the surfactant. This is a word someone invented, I don't know, 50 or so, maybe more so years ago. It's a shortened abbreviation of surface active, because it goes to the surface of that oil slick and starts breaking it up. It acts on the surface and breaks up into little tiny drops like Emily showed you. There are thousands of surfactants currently on the market. None of us buy surfactants by themselves. They're just in things that we buy. They're in our shampoos, they're in our dishwashing detergents, they're in our foods, they're everywhere. Well, there are many things. People have been making surfactants for thousands of years. I remember reading about, you know, the Romans making soap from animal fats and ash, and they're making surfactants, and you can also do it with plant oils and ash. Starting in the late 1800s, people figured out how to do this, make surfactants from crude oil, from petroleum. This is the building blocks. They could break the oil down, and the organic chemists, what they can do with oil is impressive. But there's a lot of surfactants now. That's what a detergent is. A soap is a natural-based surfactant. A detergent is an artificially based surfactant, generally from petroleum. Since the late 1800s, though, there's another way to make surfactants. And this is, you brew it, basically. You use bacteria to brew your surfactant. It's a lot higher tech, though, than brewing your own beer. These are designed microbes, genetically modified or engineered. They're designed so that they cannot reproduce. They're going to spend their life of a few days doing almost only one thing, and that's making the chemical you want them to make. You then take that chemical and purify it. You can purify it the way an organic chemist would, or you can use there are alternative techniques to purification now called green chemistry. Here are some examples of surfactants, and if you pick up some of the foods at your house or some of the shampoos or detergents at your house, you might see sodium lauryl sulfate, you might see sodium sulfonicid, I just say DOS on it. Surfactant and FA glue are two that are microbially produced that I'll talk about a little bit as well. So here's this, here's a table. I want to talk about the kind of traditional organic chemistry where you start with petroleum, you make chemicals using catalyst, heavy metals sometimes, pressure, heat, pH, and you're going to use solvents to get your product out, and then you're going to go use that product in shampoo or in core exit. With this new approach, you're using agricultural products. Glucose right now is the only way that they can do it financially compatible or competitively with petroleum, but they would like to move in and start doing it with agricultural waste products. It's a fermentation process, a single batch over a couple days, and you can use the same solvents as a organic chemistry, or you can use some of the newer green chemistry stuff. I got contacted after the spill by this company called Moleculare Genetics. I'd never heard of any of this stuff before then. They've been in business. He talked about, he's got this vision, the guy I talked to over there, Kevin Jarrell. He's got this vision that he says that one day petroleum will be too valuable to use for shampoo and detergent. One day you're going to have to use you're going to have to brew that stuff instead. You're going to brew it from either glucose, or maybe better, you're going to use bagasse from sugarcane or rice hulls, something left over that's not being used, you're going to brew it. And that's how you're going to get the fragrances and the detergents and the surfactants. Sorry, you get the fragrances and the surfactants that you're going to need to make your shampoo, for example. And so that's kind of nice. Let me know when that happens. Just know we've been in business since the 1980s. We compete against organic chemists. We sell this stuff. I can't, and he says some of our product is probably in stuff that you use. I had no idea this was going on. He says, we want to make a better surfactant. We want to design a dispersant, and we want it to be less toxic than what's on the market. I said, well, I'm kind of interested in that. Let's see if we can come up with an experiment, or something scientifically interest us. And so where we are now, EPA has put up the money for us to do some science. And so they're giving money to Iowa State University, LSU, and to Columbia University. Moleculare Genetics has given the microbes and the surfactants to all three universities to do the work on. Iowa State, what they're working on, they're trying to figure out how to make the fermentation more effective using agricultural waste products so that they don't have to use glucose anymore. What we're trying to do, and that's their interest. What Chris and I are trying to understand is the toxicity of oil and this core exit and alternatives. And so we're using this stuff to compare the toxicity of the products that they're making versus the traditional surfactants. Columbia University, those guys, they study the surfactantness of a molecule, if you will. They study how well it breaks up an oil into tiny drops of water. Or you can also break up a water slick on oil into tiny droplets. I think they do both of those things over there. But those are the three groups that are working in the project that's funding us right now. All right, so let's talk about the toxicity of some of these surfactants to fish. I'm going to talk about two petroleum-based surfactants, SLS and DOS, and I'll talk about two microbial-based surfactants. Surfactant is one of the first described surfactants, I think about almost 100 years ago. It's microbially produced. It's been used as an antibiotic. It's one that evidently Moleculare Genetics sells to people who use it in other products. They've also got this new one that they used as a base, the surfactant as a base. They said, this has got some good properties, but we couldn't use this to make a good oil spill dispersant because surfactant doesn't dissolve in water very well. So they designed some microbes to make surfactant a little bit differently and in a way that would make it more water soluble, and that's what the FA glue is. All right. The first thing I'm going to talk about is core exit. This is a list of ingredients for core exit. You don't see any of these surfactants in there, but because it's probably buried down here in this row right here where it says distillates petroleum hydro-treated light. That's like saying the rest, this is like I'm telling you the recipe for gumbo is get some vegetables and some meat and put it on rice. Vegetables could be anything, and that's kind of what this distillates here is. That's almost anything as far as I can tell. But we know that DOS is one of the surfactants in Corexit based on the research by that research group there. So Chris's group has done some work with the DOS, and they've looked at the toxicity as it's affected by salinity. And it's a very interesting pattern here, because what you see is as the salinity goes up, the toxicity of this stuff declines. Now, so at very low salinity, it takes only 10 parts per million to kill half the fish. But if you get the salinity up around 5, you've got to raise it to 30 parts per million to kill half the fish in 48 hours. And then it comes back down again. What we think we're seeing here is that the fish is trying to keep, the fish has a salinity inside its body. We all have a salinity inside our body. And one thing, it's much easier for us to maintain our proper salinity than it is a fish, because a fish has those gills and salt's always trying to get in or get out. It's got this battle trying to keep its body at the right salinity. And the salinity of the fish is up here in the middle somewhere. So as the salinity is really fresh or really salty, the fish has to work harder to maintain its salinity properly. So we think that's why we're seeing this pattern here. Very different than the pattern we saw with dispersed oil. Dispersed oil did not show this pattern. Dispersed oil is more toxic the fresher it gets. That's a little clue. One of the clues that tells me the problem with dispersed oil is not the dispersant, it's the oil part, or the dispersed oil part, but it's not the dispersant alone. But there are other things in Corexit too. There could be other things in Corexit causing that pattern. This is only one ingredient within the Corexit. Now I'm going to talk to you about another surfactant, the SLS, sodium lauryl sulfate. This is a list of ingredients in Dawn dishwashing detergent. And here we compared the toxicity of the SLS, which is the triangles on here. We use the FA glue and the surfactant. So the SLS is a petroleum-based surfactant. The surfactant is a biological one, but the natural one. The FA glue is the engineered one. Notice that very different pattern with salinity here. Implies that the mode of toxicity is not related to the osmolar regulation across the gills as it was with DOS. The saltier it gets, the more sensitive these animals are to the substance. You also see that, whoop, wrong one. You also see that, there's my red. You also see that the FA glue, the designed one, did not become as toxic as salinity increased as the other two did. So we're using the salinity of the water to help us see if the reason the substance is toxic is the same. Very different. So we got two petroleum-based surfactants here, DOS and SLS, two very different patterns in the response to salinity. Two very different ways these chemicals are affecting the fish. And then also, you know, this designed one is less toxic than the natural one, and also than the SLS, and less affected by salinity. So something's going on there. Pardon me a moment. I'm from New Orleans. I got to do that. I had to. All right. And so where are we now? Where are we now? We now are at the stage where we're comparing the toxicity of different shapes of FA glue. It turns out that FA glue comes in two different shapes. If you look at your right hand and your left hand, they both do the same thing. But they're mirror images of each other. They're different shapes. These different shapes of FA glue chemically seem to act the same. They have very similar solubilities. That's one reason why it's so hard to separate them. But we're working with some really smart people at Molecular Genetics in Iowa State who can separate these things. And Chris is starting experiments next week to compare the toxicity of these different shapes. And this is important because if we can understand, first, if there's a difference in the toxicity of the different shapes. Then we can more quickly understand the relationship between the shape and the toxicity. And then other scientists can then more efficiently design less toxic molecules that are also good surfactants. And so that's where the research is going now. All right, I've got one more slide that I'm going to show. I just added this afternoon. I almost hate to show it because it's almost a distraction from the things. I've already told you everything I pretty much want you to know. Dispersed oil is more toxic than undispersed oil. It stays toxic longer than undispersed oil in the laboratory setting. That effect is greater at lower salinity. And that's just acute toxicity. We've still got work going on with the survivors of these experiments. That's the most important thing. But people continually want to know, people are worried about this dispersant. And they've had trouble trying to put a frame of reference on it. How toxic is it? And so it'd be really nice to compare the toxicity of the dispersant to things that you're familiar with. I just found this, I'd read this abstract just before Christmas, and this afternoon I got a copy of this reprint, and I can compare it compares the toxicity of Corexit to Dawn dishwashing detergent and Johnson's baby shampoo. So here's that stuff. I'll read the words first and we'll walk through the diagram. Corexit is less toxic than Dawn dishwashing detergent. It's more toxic than Johnson's baby shampoo and Greenworks all-purpose cleaner. I'll walk you through the diagram to show it to you. So here's the Corexit over here, and so here's the concentration. You got to get up to around about 50 ppm to kill half the fish. For the Dawn is the product A. You don't have to get the concentration of Dawn as high to kill half the fish as you do for the Corexit. Here's Johnson's baby shampoo over here, product F, and here's that green detergent, green cleaner over there, project H. When I first wanted to see this answer for myself, I thought this would be a beautiful hook to get people in and say, look, this dispersant's bad stuff, but that's not what's important. What's important is the toxicity of the oil dispersant mixture and how it degrades over time. This is a kind of a distraction from that really important question. How does the dispersed oil lose its toxicity? How fast does it lose it? What conditions causes it to lose its toxicity? But people always want to know this. I got the paper today, so I'll put it at the end of this talk. And I'll come back to that, I guess. So we'll move to the questions. If Chris will come up here, we'll take some questions now. Here are our email addresses. We also have a Facebook page where we